The next portion of our program is the John K. Niparko Memorial Lecture honoring the life and work of Dr. John Niparko, one of the founders of the American Cochlear Implant Alliance. Following his passing in 2016, the board of directors made the decision to establish the Niparko Lecture to recognize Dr. Niparko's enduring commitment to cochlear implant research and clinical care and honor his contributions to the field. Given his interest in the entire continuum of treatment, lecturers may be scientists, physicians, audiologists, therapists, psychologists, educators, or others who conduct research or involved in patient care for adults or children. The first Niparko lecture was given in 2017 by Dr. Bruce Gantz, a friend and colleague of Dr. Niparko, and we have had a total of 3 lectures to date. Today's lecture is our 4th such lecture and will be introduced by Kevin Brown, a physician and scientist and a member of the board of directors.
Good morning. My name is Kevin Brown, and as Donna mentioned, I am at the University of North Carolina, and I'm also a member of the board of the American Cochlear Implant Alliance. It is my honor and privilege to introduce our speaker for the John Niparko Memorial Lecture. Dr. Tina Stankovic is currently the Sheldon and Dorothea Buckler Endowed Chair of Otolaryngology and Chief of the Division of Otology and Neurotology at the Massachusetts Eye and Ear Infirmary. In June, she will assume her role as the new Chair of Otolaryngology, Head and Neck Surgery at Stanford University. Dr. Stankovic is one of those unique individuals who define a new area of investigation and make it their own. Her prolific work in imaging of the inner ear has dazzled us with its beauty and complexity. The sophistication that she brings to imaging of the inner ear has enabled her to look at bigger questions of degeneration, evaluating markers of degeneration at cochlear synapses, and then looking at regeneration of synapses with viral vectors and drugs. I am delighted today that you will be speaking to us on degeneration and regeneration of the inner ear. So without further ado, Dr. Stankovic.
Thank you so much, Kevin. I'm deeply honored and grateful for the opportunity to give the John Niparko Lecture on degeneration and regeneration of the inner ear. My talk will have 5 parts. I will first provide a brief background, and then I will illustrate our strategy using 4 complementary approaches. As we have just heard from Donna, John was a giant in our field. His vision, dedication, and enthusiasm have been inspiring. Like the tree of life with numerous interconnected roots, John brought together different communities of physicians, scientists, patients, engineers, and technologists to advance patient care. And like the branches of this tree, which bear new flowers and fruits every season, John's legacy lives on through his trainees, colleagues, patients, and friends. John was passionate about hearing and hearing loss, and rightly so, because hearing loss is the most common sensory deficit currently disabling nearly 500 million people. And the cost of unaddressed hearing loss amounts to the astounding $750 billion annually. And prospects are not improving because this remains a growing problem. And cochlear implant has played a transformative role in addressing some of this hearing loss. It is therefore instructive to review key events that enabled cochlear implant development, because the cochlear implant is not only the most successful sensory device, but it has also set expectations for all implantable devices, because the cochlear implant involves deep understanding of the underlying physiology. It uses a number of technologies, and it is evolving continuously. So the first milestone was the birth of anesthesia, which was first publicly demonstrated 175 years ago. And this historic procedure was performed on the neck, not too far away from the ear. The second was the discovery of penicillin by Fleming less than 100 years ago, for which he won the Nobel Prize. Perioperative antibiotics are routinely used for numerous surgical procedures, including cochlear implantation. The third one was the introduction of a microscope into the operating room, which was pioneered by the 2 Swedish ear surgeons, Nylund and Holmgren, 100 years ago. And then it was the groundbreaking work of a Nobel laureate, von Bekeschi, who described the traveling wave in the cochlea and outlined the place frequency map. This was the impetus for the development of a modern multi-channel cochlear implant. And 8 years ago, Clark, Hockmeyer, and Wilson shared the Lesker Prize for their discovery and work on the modern cochlear implants. While opening a new frontier of inner ear surgery, the cochlear implant has also stimulated us to think about the current major unmet medical needs in otology and neurotology. They include 2 diagnostic and 2 therapeutic challenges. The diagnostic challenges include the current inability to see cells in a living human inner ear, as well as the current inability to routinely biopsy this organ. The current therapeutic challenges include the lack of FDA-approved pharmacologic therapies for hearing loss and the need for better devices for hearing restoration. I will now show you how we have been using the tools of basic science and engineering to address these major unmet medical needs with the ultimate goal to improve patient care. So starting with the first diagnostic challenge, I'm showing you state-of-the-art clinical imaging of the inner ear today. On the top is a computed tomography scan, which is based on ionizing radiation. And at the bottom, it's a magnetic resonance imaging scan. On the top, fluid-filled spaces are shown in gray, and at the bottom, they're shown in white. In either case, we do not see individual cells. But there are research tools that provide much higher resolution, and they include a microcomputed tomography and synchrotron radiation phase contrast imaging. So why has the ear been so difficult to image inside at a cellular level? To understand that, I'm showing a picture of the human temporal bone, and you can appreciate the coiling cochlea, which is embedded in the densest bone in the body, located deep in the base of the skull. So because of its small size, its complex 3-dimensional anatomy, and its encasement in this bone, all we know today about the cellular basis of human deafness is based on studying autopsy specimens. And in this process, the temporal bone is removed. It's then decalcified, embedded, sectioned, and stained through a laborious process that takes many months to generate a slide that looks like this, where you can see the cochlea embedded, surrounded by this purple stained bone. And you can appreciate that it's tiny because the human cochlea in cross section is the size of Lincoln's upper face on a penny. And it is also alarming that today, cochlear histopathology has been reported for only 20 out of 200 deafness-causing genes. So that means that really, for the vast majority of people, we do not know what's happening in their inner ear at a cellular level. And this slide also illustrates that deafness-causing genes are distributed across all chromosomes. But animal models have taught us the importance of degenerative processes that occur even if you have identified a genetic mutation. So in this case, I'm showing you slides from a mouse that carries a mutation in a connexin 26 encoding gene. And you can see a dramatic difference in the appearance of the cochlea on the left, which is at 2 weeks of age, and that would be equivalent of a human newborn, versus at 7 weeks of age, which is the equivalent of a human adolescence. Because by adolescence, the entire organ of Corti is gone and spiral ganglion neurons are decimated. And it tells you that if we are considering regenerative therapies based on small molecules or genes, they would be dramatically different at these 2 time points, even for the same genetic mutation. So that has stimulated us to develop new technologies that would allow us to visualize the cochlear interior at a cellular level. And we have focused on optical tools because they provide higher resolution. And I'm first showing you our approach using micro-optical coherence tomography. This is a collaborative work with Gary Tierney at Massachusetts General Hospital, who is the inventor of micro-optical coherence tomography, which is a high-resolution successor of optical coherence tomography. And in this case, we made a cochleostomy within a guinea pig cochlea and looked inside, and you can appreciate the familiar structures, including the scala tympani, the space through which the cochlear implant is inserted, as well as the basilar membrane and the organ of Corti situated on top of it. Now, if we zoom on this triangular dark space and fly through it for several hundred microns, and if you look at the bottom of the screen, you will appreciate tubular structures running across, and they are nerve fiber bundles that are headed towards the outer hair cells to modify their motility and extend the dynamic range of hearing. So now that we know that micro-optical coherence tomography is capable of detecting normal anatomy, the key question is it also capable of detecting pathology? And to address that, we used mice. We exposed them to traumatic noise levels and have demonstrated that micro-optical coherence tomography can definitely detect changes between the normal ear, shown on the top, and the damaged ear, shown at the bottom, where the entire organ of Corti has been wiped out. This has major implications for future regenerative therapies because numerous laboratories have shown that gene therapy focused on hair cell regeneration does not work in the lower case, where you don't have supporting cells that could be transdifferentiated into hair cells. Also, for other types of gene therapy, cells need to be there so that the mutation can be corrected. So inspired by these findings, we have developed a prototype probe that we have inserted into a cadaveric human inner ear through scala tympani. The probe is 600 microns in diameter, and imaging through the probe, we can see the organ of Corti. So this excites us about the next steps to really develop a probe that is targeted for human use in vivo. In the meantime, I'd like to show you another optical imaging tool that holds great promise, which is two-photon fluorescence microscopy. This is a collaborative work with Dimitri Psaltis at École Polytechnique Fédérale de Lausanne. And here we have shown that this technology allows even higher cellular and intracellular resolution, where you can see not only individual neurons shown in red on the left side and nerve fiber bundles shown in green, but also individual hair cells shown as dots on the right side. And we can detect that these hair cells are gone after noise trauma. And importantly, this tissue was not stained in any way. The color represents intrinsic optical properties of the tissue. When considering using of two-photon or multi-photon fluorescence microscopy applied to the human inner ear, right now it's much easier to consider a rigid configuration of the endoscope than a flexible one, which would really limit our imaging to the very base of the cochlea. So for that reason, we have considered another option, which is to shave down the cochlear bone at a few strategic locations to make the bone thinner so that we can look through it. And in this case, we used a femtosecond laser to very precisely shave off tens of microns to make the OD capsule thin enough so that we can then look through that bone while the inner ear remains intact. And then we can appreciate inner hair cells which had previously been labeled. While developing these new optically based technologies to allow future cellular level interrogation of the human inner ear, we need to have a method that allows us to validate our approach. And I've already told you that the standard histopathologic examination of the human inner ear is a lengthy process that takes many months, so it's not practical. And for that reason, we have identified synchrotron radiation phase contrast imaging as a very promising tool for rapid interrogation of the human inner ear, and in particular autopsy specimens. And in this slide, the image was obtained without decalcifying the specimen and without staining it in any way. And you can appreciate the beautifully spiraling structure of the organ of Corti. And in fact, if we make a digital section at the level of the organ of Corti, you can now appreciate the spiraling mosaic of cells. So with that, I would now like to move to the second diagnostic challenge, which is the current inability to routinely biopsy the human inner ear. Tissue biopsy of this organ is impractical because the organ is so small that tissue biopsy would destroy it and thus cause hearing loss. However, there is a great potential in biopsying the liquid within the inner ear. There isn't too much of it. There is the equivalent of 3 raindrops of fluid in the human inner ear. However, if we take a small fraction of it, then liquid biopsy may be feasible. So to enable that, the first step that we did was we described the proteome of both normal human paralymph and diseased human paralymph. And then we developed a device for controlled and reliable cochlear liquid biopsy. This is a very simple device. It's just a beveled microneedle with a step-off that provides a mechanical safety feature, and that there is an additional electrical safety feature that prevents trauma to the inner ear from overinsertion. We have tested this device in human cadavers, and on this picture that's shown in blue, you see a little hole that was made by the device, which is only 250 microns in diameter, which means that it can either heal readily spontaneously or after application of a small tissue graft. And it could enable routine liquid biopsy of the inner ear in the future that could be repeated. But an important question to address is it possible to detect molecular biomarkers of hearing loss in a tiny amount of fluid? And the answer is yes. We have shown that in as little as half a microliter of inner ear fluid, we can detect molecular biomarkers of hearing loss, which really paves the way for future routine biopsy of the human inner ear to guide diagnosis and therapy. That now brings me to the first therapeutic challenge that I would like to address today, which is the lack of pharmacologic therapies for hearing loss. And we have focused on drug repositioning or repurposing to begin with, because it has many advantages when compared to the traditional drug development, which has a high failure rate of up to 90%. It's a lengthy process taking up to 10 years, and it's super expensive in excess of $2.5 billion. In comparison, drug repositioning has low failure rates, especially when it comes to safety. It's relatively short and considerably less expensive. And I'll show you how we have focused on one class of drugs, which are called bisphosphonates, and which are used to treat bone diseases. So that begs the question of what's the link between bone and sensorineural hearing loss? Well, this question was inspired some 15 years ago by the work of Mike McKenna's group, where they showed that osteoprotagon is abundant in the inner ear fluid. And as the name says, this molecule, osteoprotagon, protects the bone from remodeling. It's really interesting that it's expressed in the inner ear at levels that are an order of magnitude higher than in blood or cerebrospinal fluid. And we were intrigued to realize that it is cochlear neurons that express this molecule at the highest levels in the inner ear. So is it possible that these neurons are expressing this molecule simply to prevent the surrounding bone from remodeling? Or is this molecule also important for sensorineural function in the inner ear? And to address this question, we used osteoprotagon deficient mice. And we have shown that these mice develop sensorineural hearing loss in addition to the conductive hearing loss due to malfunction of the middle ear bones. And this sensorineural hearing loss is characterized by degenerative changes within the cochlear nerve, as well as premature aging detected by electrophysiologic testing. We then showed that these osteoprotagon deficient neurons are sensitized to oxidative stress, which mimics acoustic trauma, as well as programmed cell death, and that floating spheres or stem cells derived from the spiral ganglion in these mutants have reduced proliferation and survival. And if you look at these images at the bottom, you will see on the right what looks like sand or dust, which is very abnormal compared to normal looking spheres on the left side. And importantly, this phenotype could be rescued by the addition of zoledronate, which is a bisphosphonate. Well, this works for osteoprotagon deficient neurons, but now the question is that relevant for normal cochlear neurons? And to answer that question, we exposed mice to noise levels that cause selective loss of synapses between inner hair cells, which are shown in blue, and auditory nerve fibers shown in green. If you now focus on these green fibers, you will appreciate that they start disappearing after the appropriate noise levels are used. So we use such noise levels and in mice, and then measured the auditory brainstem evoked response, and in particular, the magnitude of the first wave of this response, because it's the most sensitive metric of neuronal integrity in the inner ear. And we find that after noise trauma, this wave 1 ABR amplitude is dramatically reduced, shown in the gray arrow. And after the addition of zoledronate, which is given after acoustic trauma, we could rescue this sensorineural hearing loss. And this physiologic response was corroborated by the histopathologic demonstration of regeneration of synapses within the inner ear. And you can appreciate that on the top image in the middle, where there are many more of these red-green pairs, which are the pre- and postsynaptic pairs, compared to the bottom image, which is after noise exposure. Now, what's important to consider here is that these drugs can have significant systemic side effects. So that begs the next step, which is localized delivery to the inner ear. And one way to do that may be via a cochlear implant. So now I'd like to move to the final therapeutic challenge that I'll discuss today, and that's the need for better devices for hearing restoration. We all know that conventional cochlear implants have an external component and an internal component, and that there is a need for fully implantable cochlear implants, not only to address the social stigma and cosmetic consideration, but also to improve function. So working with Ananta Chandrakasan at MIT, we developed a prototype chip for a fully implantable cochlear implant. And the key advantages of our system include, one, the sensor in the middle ear, which is a piezoelectric sensor, so that it detects vibrations of the middle ear bones just the way we normally do. The second feature is a highly reconfigurable sound processor. And the third feature is an energy efficient waveform. And we tested this energy efficient waveform in cochlear implantees and found that it amounted to significant, up to 25% savings in both charge and energy. Since then, very important work has been done by others on enabling TICI, or the totally implantable cochlear implant, and was inspired by the initial work out of Australia, when in 2008, the initial experience was described with the TICI research device. Importantly, a few years prior to that, a patent was awarded in the United States for a fully implantable cochlear implant system. More recently, 3 years ago, going back to Australia, TICI clinical feasibility study was furthered by the Cochlear Corporation. And then last year in the United States, the FDA granted a breakthrough device designation to the acclaimed Cochlear implant, which is a fully implantable device. And then just a few months after that, in Europe now, the first sticky surgery was performed in a clinical feasibility study testing a Medel device. So the future is definitely bright on this front. And it begs the question of how are these totally implantable cochlear implants going to be powered? Because every battery has a finite lifespan. And we have considered the possibility of powering some of the devices by the body itself. And working with Ananta Chandrakasan, we demonstrated for the first time the feasibility of extracting energy from an electrochemical gradient in the body. And in particular, we extracted energy from this tiny biological battery in the inner ear, which is called the endocochlear potential. And we used this energy to power a sending antenna, allowing transmission of information to a receiver located a meter away. And while we don't think that cochlear implants and hearing aids of the future will be entirely powered by the inner ear, it brings the possibility of future hybrid devices partly powered by the body. So what is the future? I think it's very promising for cochlear implants, and we can consider it in terms of two flows. One is the flow of information, which has been the traditional focus of research, but another one to consider is the flow of energy and matter, which will enable better integration with the body. And both of these flows can be achieved through improvements in hardware and software. When it comes to hardware improvements, there is clearly a need for more electrodes and better signal localization at the interface. And this cannot be achieved simply by electrical stimulation because of the spread of current that we are all very aware of. So future promising options include optical stimulation via optogenetics or infrared stimulation, as well as magnetic stimulation. Another opportunity is for better tissue interface, such as with nanocoating that provides nerve fibers to reach and interface with the device, as well as immunocoating that would prevent an immune reaction and a foreign body reaction to the device. Another opportunity is a better body interface. I've alluded to the possibility of energy extraction, at least partly from the body, to prolong the life of an implanted battery, as well as the possibility of controlled drug release. Now, when it comes to software development, it includes the possibility of artificial intelligence being used for speech processor tuning, as well as using a cochlear implant as a sensor, not only of the ear, but also the nearby structures, because the cochlear implant has a prime real estate. And in fact, recently a paper was published demonstrating for the first time the possibility to detect and measure an electroencephalogram from a cochlear implant electrode array, which really opens a new avenue altogether for neural monitoring, continuous neural monitoring, and for neurotheory prosthesis of the future. There is also an opportunity to address fault accommodation of cochlear implants via telemetry and continuous monitoring of not only all of the electrodes, but also of monitoring of the energy flow. And ultimately, when we are talking about people with multiple implants, not only cochlear implants, but brain implants and retinal implants, then there is a very interesting possibility to consider energy sharing among these implants and to consider sharing of communication among them. So cochlear implant indeed occupies the prime real estate as in it, and is a natural hub for such a system of implants. And exploring all of these options will be in the spirit of John Niparko's work, because he always had patients' best interests in mind and demanded from his collaborators that no stone be left unturned when looking for options to solve challenging problems. In summary, I've shown you our approach towards personalized diagnosis and therapy of hearing loss, which is largely inspired by the success of cochlear implants and opens new avenues for cellular level diagnosis and therapy of sensorineural hearing loss. And this is a part of a bigger framework of precision health that is the strategic direction of Stanford Medical School, where I'll be moving to in a month. I believe that our field can contribute to this approach in a major way because we have been quick to adopt and develop new technologies while straddling medicine and surgery. I would like to thank people from my lab who have contributed to the work I've described, and my many collaborators across many institutions in the world. And I'm deeply grateful to our funding agencies who have supported our work over the years. Thank you.
Thank you, Tina. That is absolutely terrific. I think, as much as we sometimes sit in awe of the miracle that we are able to accomplish with cochlear implantation, I think you've really highlighted that where we're at is just the beginning, that there is really an incredibly bright future moving forward with technological advances in cochlear implantation. As is tradition for the Niparko Memorial Lecture recipients, Dr. Stankovic has already received her bowl. This was from a favorite store of Dr. Niparko and is accordingly inscribed.
Yeah, thank you again so much, Kevin and Donna and the American Cochlear Implant Alliance members for this humbling honor.
Thank you again, Tina, and thanks to everyone for their attendance. And with that, I will close this session. Please feel free to move to the next. Thank you.